Hi, everyone. Thank you so much for joining us for the fifth webinar in the NECAN Industry Webinar Series. I am Meredith White, Head of Research and Development at Mook Sea Farm, which is an oyster farm and hatchery in Maine. I'm also chair of the NECAN Industry Working Group, which has put together this series. This series is designed to share information on how changes to coastal ecosystems, including ocean acidification, are affecting aquaculture and fishing industries. Today, we will hear from Bassem Allam about climate change impacts on the health of commercially important shellfish species. Bassem's presentation will be about 45 to 50 minutes, followed by a question and answer session. You're welcome to submit a question during the presentation. If you do so, please indicate what aspect of the presentation you are referring to, and we'll wait until the end of the presentation to go to the questions. Bassem is the Marinetics Endowed Professor at Stony Brook University and the Director of the Marine Animal Disease Laboratory. He earned his PhD from the European Institute for Marine Studies in Brest, France, and performed his postdoc at Rutgers University in New Jersey before he joined Stony Brook in 2003. His research centers on the physiology and host microbe interactions in shellfish, the resistance of these animals to infectious diseases, and how the environment affects these interactions. His work has resulted in the publication of over 80 peer-reviewed scientific publications and book chapters on various aspects of shellfish physiology and pathology. And with that, I will turn it over to this presentation. So thank you so much.
Hi, everyone, and thank you, Meredith. Everybody's hearing me fine, Meredith? Yeah.
Yes, we can hear you.
Correct? Yes, okay, perfect. Hi, everyone. So, yeah, I'm Bassem Allam. As Meredith mentioned, I run the Marine Animal Disease Laboratory, and I will be talking today about some of the studies we have been doing over the last 10 or so years on various species and how they are impacted by microbial pathogen and how the environment is impacting these interactions. But first of all, I would like to introduce the small unit that I run, the Marine Animal Disease Laboratory, which is a research and diagnostic facility that was established by the New York State Legislature to perform research and diagnostic and other related activities on the occurrences, causes, and effects of marine animal diseases. This comes from the legislative text that formed the lab. So indeed, we do lots of research on various aspects of animal health, including the physiology, the pathology, the immunology of these organisms. In terms of diagnostic, we provide service diagnostic to stakeholders, mostly state agencies, but also aquaculturists in New York and throughout the Northeast. And here are the slides show some of the species we work on. So the focus of today's presentation will be on the climate and how that climate change impacts these species. And I like this picture because it shows most of the alteration we've been seeing in the marine environment as a result of climate change. We have change in salinities, acidification, and increase in temperature. Basically, we're cooking planet Earth. The only thing missing here is really the sea level rise. Although that's a very important aspect, it's less impactful on the examples I will be talking about today. So these climate alterations have really broad and profound impacts on the physical-chemical characteristic of the marine environment. We are changing all these parameters, and this is impacting the species we are interested in. And I presented here a few graphs that shows what I mean by that. For any given environmental factors, any animal will have an optimal range, right? Beyond which, whether that environmental factor kept increasing, say salinity, for example, in a given amount of salinity, the organism will do very well. If the salinity is below that level, the organism will be stressed. If the salinity is higher than that level, the organism will also be stressed. So think about it as optimal range for any given species that can impact physiology. And when I say physiology, I mean any aspects of physiology. This is what is meant here by any biological metric. Could be growth, survivorship, reproduction, immunity, resistance to infection, and so on. So really think about it as optimal range, and above and below, it's not good. And so we have all these changes going on, and all of these physiological processes being impacted. So it's a little complicated mechanism here that we need to look carefully at what's going on. So the outline of this talk is really, instead of focusing on a species or a couple of species, I felt that the audience might be very diverse, and I thought that we can talk about a few more examples on species we've been studying on and off over the last few years. So I will cover really some aspect of the lobster health problem in southern New England, some studies we did on the immunity of these organisms. I will be mentioning the spread of oyster diseases in the Northeast. Some investigation we did on the soft clam decline here in our region and for the south. But also, you've heard previous presentation in this series of webinars, but also otherwise, about the huge impact of ocean acidification. Today, I'll be talking about some aspects that are less investigated so far, namely the effect on immunity and resistance to infections in these organisms under ocean acidification. And I will finish with a small positive note on some rather encouraging finding we found on a particular disease in hard clams under changing environmental conditions. So with a positive note at the end. Let's start with the lobster, the American lobster. This slide here shows, although it should be animated, I don't see the animation on the screen, that's fine. It was a GIF animated initially. Nevertheless, the distribution of the lobster, and we see when the animation works, although it's not working, that the species is really a little shifting north in terms of population density. So currently, Maine's, for example, landings and population estimates have increased dramatically over the last 20 years or so. We are talking about 200% increases in Maine and a decrease in southern New England. Again, I apologize. I had it working on my PowerPoint alone, but not on the WebEx system here. Nevertheless, so the point here is to tell you that this species distribution, the abundance of lobster are decreasing in southern New England, southern Massachusetts, and around Long Island, particularly Long Island Sound, and increasing further north and northern locations. This is not the species, the individual that are migrating, of course not, but it's a matter of recruitment and mortality causing an overall shift of the population. And the hypothesized cause of that is truly environmental change, in particular temperature. This map here on the lower, the bottom shows the change in temperature since 2004, and the red tells us it's about 0.2 degrees centigrade on an annual basis, increase in temperature. So here, if you look at the Northeast region, we are in the dark red. It's in the 0.2 or a little more even degrees centigrade a year increase over the last 15 years or so. So that's pretty striking, and in particular since 2005. If you see here on the top graph, this curve with an significant increase in temperature over the last few years. So we have a huge increase in temperature, fast increase lately over the last decade or so. So a little closer to us, to New York here, this is in southern New England, a study done in Rhode Island that showed distribution or estimate of abundance of lobster between 1990s and 2012. And as you can see on the bar graphs, there is a significant decrease in abundance associated with a 2-degree increase in summer surface water temperature over the same period of time. So the trend is confirmed in this study as well. And this is something we see in landings. This is an example here in Long Island Sound. A Long Island Sound landing that really peaked in the mid to late 90s. In 1996, I believe, was the highest landing. And we had a significant reduction in landing in the following next year, in particular associated with a huge mortality event that happened to lobster in 1999, in the fall, late summer and fall of 1999 here in Long Island Sound. I should say that Long Island Sound is considered the most southern inshore fishery for the species. So again, we have lobster offshore, but inshore, it's a more confined environment where environmental condition can be significantly different from offshore, more deep water. So this is the most southern fishery, and it really crashed. As you can see on these the trends, up to now, there is no recovery in lobster population. So what happened really? Well, there is lots of discussion about what happens during that mortality event. The truth is that lobster were being pulled dead from traps, so they were really dead. Severe mortality event that happened that late summer and fall of 1999. This is a picture of some of the catch that were in traps at that time. It's a really impressive severe mortality of lobster. And there were a lot of questions. What happened really in that mortality event? As you can see here from newspapers clips, it's stirred lots of debate, even lots of discussion still ongoing until today. There were pesticides suspected. There was even court settlement done. But the truth is that when you look at environmental data, this is how the data looked like. The mortality event in 1999, this is the first red bar here, follows a whole year of positive temperature anomalies in Long Island Sound. So the temperature in Long Island Sound was warmer than usual throughout the year, and indeed it continues over a couple of years, as a matter of fact. Since January 1998 here, up until the mortality event, all temperature measured were higher than average. And data collected by the Connecticut DEP showed some of that. This is the trend here in September. This is the date here of the sampling, September 1 to the 3rd of September 1999. And we have temperature reaching 23 degrees in the western part of Long Island Sound, where the mortality was most severe in the western part of Long Island Sound in that mortality event. Even throughout the whole sound, we had a temperature up to 22 degrees centigrade. That's certainly at the far end of optimal temperature for lobster. Yeah, it's really, when you look at most papers for adult lobsters, the tolerance limit is considered around a little above 20, between 20 and 21 degrees, depending on the studies. So we were throughout the whole system above that temperature. So we are pushing already, if you remember the curve on the second slide I showed, the curve, we are pushing the lobster toward the far end of that curve, yeah? Way beyond the optimal range. And so what would happen in this case? Well, we did a small study with the colleagues here in Stony Brook, and we showed that there is a crash in immunity as measured in this particular graph by what we call phagocytic activity of blood cells, meaning the ability of blood cells to engulf and degrade microbes, which is their role, basic role, is significantly decreased in lobster held at ecologically relevant temperature, 23 degrees, as compared to a more optimal temperature of 16 degrees. So we have complete crash in immunity in these animals. So the question is, what would happen if immunity decreases? It happens the same in human, in any animal. If immunity decreases, we can have opportunistic pathogen flourishing and attacking the host. And this is pretty much what was seen at that point, when virtually all lobster dead or dying from that mortality event were infected by amoeba-like parasite that was killing the lobster, suspected at least to be killing the lobster. And that amoeba, interestingly, was not known to be a pathogen to lobster before that date. So it's really forcing the system, forcing the environment to a level that is stressful to a degree that can lead to opportunistic infections. So lobster, it's not the only problem we had with lobster here in the southern New England. We had another severe infection caused by microbes called shell disease or epizootic shell disease, to differentiate it from impoundment shell disease. So this is seen on the carapace, as this picture shows, with some holes like cigarette burns on the shell, can completely degrade the shell, reaching the meat underneath. So the infection can be really severe, leading to mortality as well. This graph here, done by Cathy Castro at URI and published in 2012, shows basically the abundance of lobster here in the solid line and shell disease prevalence. And we see that the two curves crossed each other here, which seems to indicate that at least some decrease in population abundance may be related to increase in levels of shell disease. So we may have loss of lobster because of the disease and mortality. So looking at the trends, the temporal trend here on the map, so the bigger the red dot, the more disease we have. And you can see that the disease has been increasing over the last couple of decades in southern New England, really reaching up to over 30% in some spots here, off Rhode Island, for example. So shell disease can lead to decrease in abundance, and it's on the rise. And this table, I can summarize the same data, with the hot spots here in southern New England being eastern Long Island Sound, Ellis. Western Long Island Sound, interestingly, they didn't have much of shell disease, as you can see in this column. Rhode Island, we had a hot spot, clearly. Again, the values over the 30% we see at the end. Southern Massachusetts as well is being hot spot. Northern Massachusetts, Boston Harbor, low levels. And Maine is a low level as well, but I will get back to this later in the talk. So what's going on in southern New England? Why you have such high level of disease? We took the hypothesis that these animals might be stressed somehow, leading to a decrease in immunity. So we did investigate their immune processes, and not wanting to go too much detail about what we measured, think about it as being measuring, well, antibacterial activity in the blood. This is an enzyme that contributes to the killing of microbes as well, and melanization, phenol oxidase activity. Phagocytic activity, I mentioned it earlier, ability of blood cells to engulf and degrade microbes intracellularly. And another antimicrobial process, we call it oxidative burst. 4 different immune parameters. And systematically, the lobster from eastern Long Island Sound showed the lowest levels of these 4 immune parameters. And considering all data together for all these immune parameters on what we call principal component analysis, clearly we distinguish lobster from eastern Long Island Sound from lobster from Maine that had better immunity, and from lobster from western Long Island Sound, which had also better immunity. Interestingly, because we by default consider western Long Island Sound to be more stressed environment. Nevertheless, eastern Long Island Sound had really the lowest immunity, and we think that has been related to enhanced prevalence of shell disease. The study, actually, the paper compare infected and uninfected lobster as well, but I'm out of time to talk about much detail on that. The manuscript has been published a few years ago. There is another joint manuscript on the microbiology of the lobster. If you're interested in those, I encourage you to go on our website and get the full papers. Let me go back to the shell disease trends in Maine. I said, indeed, they were relatively low, still below 1%, way below 1% even. Although a more recent study, this study published in 2012, so the data is at least a year or two ahead of time, before that. A more recent paper published in 2018 showed a significant increase in shell disease in Maine, in particular in the western regions of Maine, and up to 22% prevalence. So 22% of very large lobster specimens had epizootic shell disease. That's very, very high level. And another somewhat concerning study was also published in 2018 that linked increase or worsening of shell disease symptoms and prevalence with increase in temperature. So the higher the temperature, the more disease development is expected or demonstrated in this particular study. And going back to my earlier slide about the trends in temperature in the Northeast and in the Gulf of Maine in particular, that's somewhat concerning indeed. As for Long Island Sound, the fear here, I mean, the community around here fears clearly that lobster is gone from Long Island Sound. I mean, there are still some, but the fishery vanished really. Very small effort of fishery is still going on, and it's not began to say. And the trends are also concerning in the sense temperature is increasing, as you see on this graph. For bottom temperature and surface temperature, they follow almost parallel trends, and they are both increasing gradually and continuously. So it's a big concern indeed for a fishery like lobster. Switching gears, so lobster, let's say lobster is a cold water species. Yes, it will be stressed under changing environmental conditions, under warming trends. Let's take another species. It's the eastern oyster here, which is considered a warm water species. When you look at the distribution, actually it goes in between central, and actually I didn't show on the map here, the species goes down to Brazil. There are Crassocerea virginica population in Brazil, but the species flourishes really in the Gulf of Mexico, in the mid-Atlantic, at least historically, large population. And it's present, we have wide population in the Northeast as well. So that's the species, rather warm water, so one would expect it to do better with warming trends. And that's not the case. And actually what we see is, for example, this data from New York State here in terms of landing. This is landing. And we have clearly a clear dip in production. And on the graph here, I added the few pathogens that impact the species. Eastern oyster population are heavily impacted by infectious diseases, and actually the population health as a whole, the main driver for those population health are considered to be infectious diseases. Of course, we have overfishing on one hand, but otherwise, in terms of natural factors, diseases are the most important factors regulating population. So here, MSX, one of the earliest disease, we don't know exactly when it started, although we have a good sense when possibly it started. We know it started in Delaware Bay, but when it reached here, our area in New York, we are unclear. We have juvenile oyster disease that appeared in the late 80s and early 90s, and dermo, otherwise known as Porcensis marinus, that is appeared here in the early 90s, but I should say that this parasite originated initially from the Gulf of Mexico, at least discovered in the Gulf of Mexico. The parasite is shown here on this picture in oyster meat. But the geographic distribution of the parasite is interesting because it was discovered in the Gulf of Mexico, then known to be really up to lower Chesapeake Bay in the 80s. By the early 80s, we knew it is up to Chesapeake Bay, lower Chesapeake Bay. And then in the 90s, really made a quick jump, almost 500 kilometers north. And to make a long story short, several studies were done. I'm showing here the two of the earlier studies that clearly link that spread in the Northeast for Porcensis with increasing temperature in the water, particularly winter temperature, that were apparently the limiting factor for the persistence of the parasite. The harsh winter killing the parasite or limiting its spread, and when this winter became a little milder, the parasite was able to sustain itself and killing the oyster the following year. Yeah, we have here temperature data basically in terms of anomalies. Zeros on this graph represent the average. Anything below zero is cooler than average. Anything above zero is hotter than average year. And they are cumulative, so we can have several years in a row, and this add up on the curve. So again, clearly the spread north happened after extended periods of warmer than average winters. Milder than average winters. That's been a fact really, and the lab experiments actually support that. This is again very early work, not out of our lab, by Xu and Lapierre out of VIMS, showing that the disease, Perkensis marinus infection in oyster, heavily depend on temperature, with a very low development of the disease at 10 degrees opposed to 20 or 25 degrees in the study. And another thing that is somewhat the good news, the parasite seems to be somewhat limited, not completely eliminated, but somewhat limited at low salinities. So in these studies, they tested 3 practical salinity units, 10 and 20, and the disease is lighter and low prevalence at the low salinity and higher at the high salinity. And this is a good thing because we have oyster population that can survive in low salinity places with little impact of the disease. So these what we call disease refuge areas. We have for Perkensis marinus, we have disease refuge areas in low salinity, but also for MSX, the parasite I mentioned earlier, disease refuge areas in low salinities as well. So here we looked at those disease refuge area and how they could be impacted by variation in salinities. The study site here, this is done and led by my colleague here in Stony Brook, Professor Jeffrey Levington. I provided help and assistance with mostly with disease diagnostics, but the study really focused on the Hudson River estuary. And in particular, this area here in somewhat upper Hudson River, yeah. And in particular, this site here that is called Ossining, OS. You'll see it in the next few slides as OS. So this is really the highest point here on the map. So considering it's a river that goes to an estuary, the salinity, there is a nice salinity gradient for marine offshore estuary in here in the 20-25. And going lower as you go upriver. So we, Jeff, under his leadership, deployed oysters in these different places and followed their progress. And the good news is that, yes, we have indeed disease refuge area in these Hudson River estuary areas up the river here. So this is disease prevalence. The higher the worse prevalence it is. So we have very low prevalences in upriver, up Hudson River. This is the salinity on the x-axis. So we have a few sites here with very low disease because it's low salinity. Indeed, the oysters do not grow as well. This is growth and salinity. They don't grow as well, but they are protected from disease. So that could serve as a refuge area. And oyster can sustain themselves there and reproduce even slower, even take longer maybe to reach maturity and reproduction. That's fine, but they are protected from the disease. Well, what we noticed is that even in those protected areas, we can have mortality and severe mortality. This is a study done over 2 years. This is finding in 2008, the year here on the x-axis, and the date, the day of the year, basically. So the Ossining site I mentioned, that was the highest on the river, OS here in brown. We see that we have mortality that happened in the summer of 2008, and severe mortalities that you see here on the right hand, the brown curve again, up to 100% mortality of oysters. And this is no disease, not related to disease. What is the reason? Was freshets. A heavy import of fresh water into the system. And this is the salinity here on the second series of the curves, salinity. measured in 2008 in the first on the left-hand side, and in 2009. And look at the salinity level that we reached in 2009. The brown curve here on the salinity, we did hit the zero salinity for extended period of time. And actually, throughout most of the summer, I'm talking over a month, the salinity was consistently below 3 parts per thousand. That's low. That's very low, and reaching really near zero or near zero for a very extended period of time, we think was at the origin of this drop in survivorship. And interestingly, the two other sites in the same bay there up the river, these also upriver sides, have also high mortality. So clearly, this is a input related to fresh water and lower in salinity below the range that is acceptable by the oyster. The last graph here, at the bottom graphs in 2008 on the left-hand side and 2009 on the right-hand side, shows actually the river flow on the y-axis, and we see nice peaks here in river flow, particularly in 2009. A nice peak in river flow associated with a significant drop in salinity and with the mortality event. So we were able to link in this study really the mortality to drop in salinity that is a direct result from precipitation. The most worrying part is the projection about precipitation here in the Northeast. As a matter of fact, we expect precipitation to increase by up to 25% over the next 8 years or so now. An increase in precipitation by 25% will increase, of course, the river discharge, and an increase in the river discharge will lead to much lower salinity. So we will be hitting that 0 to 2 parts per thousand more often for more extended period of time. And that's a fear, because those refuge areas upriver, where we know there are some natural oyster populations, can be lost or at least shifted further south down the river. So that's a concern indeed about projected change in this particular case. So the third study, really I want to mention, the third case I want to mention is surf clam. This is a very important fishery here in the Northeast, and in New York in particular. It's still the first fishery in New York State in terms of landed biomass, although we see a decline. This is the landed biomass over the last 15 years or so, 14. With a decrease overall, but still one of the most important fisheries. And this is not specific to New York State. This is we have here in New Jersey a decrease between 1997 and 2004, and the Delmarva Peninsula as well, decrease. So it's a general trend. And what we see in New York, this is data from the state DC, we see that this decrease in biomass is associated with the loss of the juvenile. We have the size here on the x-axis, and these juvenile that we've been seeing early on in 2002 survey kind of disappear, and we don't see that much new recruitment happening on in the following surveys, 2008, 2012, for example. So that's a big concern about the health of the population. And we had a student here in Stony Brook in our group who did analyze the population data with prevailing environmental conditions and was able to kind of still correlation at this point, and this is something, a work in progress, I should say, correlate recruitment events with relatively cooler than average years and lack of recruitment in those hot years, hotter than average. Again, 0 represents average. Below 0 is cooler than average, above 0 is warmer than average, and the arrows represent recruitment events that we can determine from the population surveys. So Jesse Hornstein was able to really link those recruitment events to cooler than average years, to cool years instead of warm years. And this is something that has been strongly suggested in the literature. I mean, there are previous studies, there are a few studies that link or at least strongly suggest that the change and the shift in the populations of surf clam is related to increase in temperature in the region. So I will only talk about a study that Jesse did in parallel to those population work. He did a physiology study to evaluate the effect of temperature on surf clam physiology. And what he demonstrated was that significant change in physiological parameters were shown in surf clam exposed to high temperature. In particular, things like excretion. So higher excretion in surf clam exposed to high temperature, less ability to assimilate food, ingestion rate decrease, condition index decrease, and so on. And most important, I think, the scope for growth, which means all energy balance available for the different physiological functions was significantly lower at 23 degrees as compared to the control, which was in this case 19 degrees centigrade. So the temperature was extremely stressful to these animals. Again, this has been shown also by Jesse in terms of energy reserve in the meat of the surf clam, in terms of glycogen in particular, even in terms of protein and muscle, but he did also the mantle, so also decrease in glycogen and in lipids. So clearly these animals at 23 degrees started burning their energy reserve to function. Yeah? Or at least are unable to accumulate as much as control animals held at a more optimal temperature. And this is also seen in the immune abilities of these surf clams. For example, the phagocytic activity, again, I mentioned this earlier, ability of blood cells to neutralize foreign microbes is lower in these animals held at the higher temperature, and they succumb more easily to infection by bacteria. This is all experimental work done in the lab, but has implications for the field, especially that these temperature can be reached not in the bottom water where surf clam are present, although they can be occasionally, but the risk is that we can see them more and more often down the line. And actually this stress, temperature stress, we've seen in the lab experiment and in the field observation that these animals can start degrading their gonad tissue before spawning. So because they are stressed, they can start using the energy reserve stored in their gonad without releasing these gametes and spawn. This is obviously have a major implication for the population health overall, but also for the individual. So the individual started using their gamete material to function metabolically. That's we see. This is a work in progress, I should say. I mean, we have strong evidence, but the work will be continued this year and next. So shifting gears a little, and I try to keep this part relatively small because, again, you've heard a lot about ocean acidification. I think some studies showing, well, and for some species we have positive effect of acidification, although there are only a few. In many cases, it's negative. For bivalve mollusk in particular, it's extremely negative. Most studies were focused on shell growth, calcification, on the survivorship of the animals as well. In some studies, we had what was called, at least by others, neutral effect. That means effect not seen, basically. Not seen doesn't mean it doesn't exist. Could be just the parameter that were measured in the experimental condition used did not allow the observer to see anything. Doesn't mean there is no effect, of course. And one aspect that we focused on in our group here is the immune processes, how acidification affects immunity. And I will show you only a few slides. The work is still ongoing, and we have a couple of students interested in these aspects. who are working on really formalizing the first data sets for publication. But quickly, this is a first slide that focused on oyster larvae, so the Crustacea virginica, the eastern oyster, that were exposed to either ambient partial CO2 level or elevated. The normal, the ambient was, I believe, 400 target, and the elevated was, I think, my recollection, 1,000 ppm PCO2. So exposing these larvae to these two different conditions, high PCO2 and normal PCO2, and then exposing the animals to bacteria or not. This is what is shown here by bacteria. So 24 hours, and we have two experimental design, either maintaining the CO2, partial CO2 pressure stable by bubbling CO2 into the tank, experimental tanks, or, sorry, no bubbling here, or bubbling on the right-hand side. So two different experiments, actually, with slides. The first experiment, we didn't bubble CO2. So the animal were raised at different CO2 level, but during the exposure to bacteria, the CO2 was not bubbled, so there is a shift in the pH probably during that experiment. Nevertheless, we still see that the animals that were exposed to acidification were the only one that showed mortality following exposure to bacteria. Those exposed to bacteria, but at the ambient level of PCO2, did not display any mortality following bacterial exposure, the green bar here. The same similar trend, or at least more pronounced, was in the experiment where we did continuous bubbling of CO2, where bacterial challenge was the highest mortality, almost 100%, although it's maybe 97 or so % mortality, when the larvae were exposed to CO2 and then challenged with bacteria. And then we have ambient with bacteria following elevated without bacteria, and lowest mortality in the ambient without bacteria. So a nice gradient here that clearly show that adding stress on stress, bacterial stress on pH stress, worsen the situation, obviously. So the animals are trying to already combat the pH stress, and then that weakened their immunity and resistance to infection. The same, we found it in clams. Again, a nice range here with, yet again, the highest mortality were noticed in clam larvae exposed to low pH and bacteria, followed by ambient with bacteria as well. So the same kind of trends here over time was noticed as well. This is a more longer-term experiment where the clams were grown for over a year, under or about a year, in acidified or not condition, what we call ambient A, and then moved after that year to either elevated, in that case we have letters ambient to elevated, A, E, like here. If you can see the third series of the bar graph, you don't see my mouse, or kept under ambient. A means were in ambient, kept in ambient. EE were in elevated PCO2 and moved to elevated CO2, basically kept in the same place. Or EA, elevated, moved to ambient, and then exposed to bacterial challenge. The bacteria are the red bars. And we see actually the clams too kind of recover rather quickly, even after a year of exposure to acidification. Moving them to ambient, they can rebound and resist infection. The same for the elevated. Keeping them at elevated, well, they already are hit somewhat hard. So adding the bacteria in this case, it didn't make much difference. But the most important, I think, part of the story is that rebound and elevated that were moved to ambient, and then they did fine in terms of control. Of course, bacterial challenge still have a toll on them. So again, this is what I meant by still really a black box. The effect of acidification on immunity, still not many studies did to focus on that. We have no knowledge whatsoever of the effect of acidification on the parasites and pathogens themselves, although we started working on that as well. Because again, we are looking at the interaction. What matters is the whole interaction between the host, the pathogen, the environment, and how all these impact each other. So it's more complicated than just calcification or survivorship of larvae. So to finish, I really wanted to talk about something optimistic at the end. And the only thing I can think of with regard to climate change and positive things in marine invertebrate is really this particular case, the hard clam, and not the hard clam in general, because we saw in the previous few slides that they are impacted by acidification heavily, and that which can worsen even their resistance to infection. But here we are focusing really on a disease we call QPX disease in clams. And this is, I'll cover it in the next few slides. Again, this is a beautiful picture, I think, that was found by one of our previous students, Long Islanders, the inventors of ice climbing. I don't know if anybody else in the Northeast do ice climbing, but this is out of Great South Bay back in the good old days of hard clam fishery in that bay. So the parasite, QPX disease, it impacts hard clams. It's a fatal parasite, can lead to virtually 100% mortality, but also can be very low levels in some conditions. And it's important to understand those conditions. So the pathology of the disease, really, what we see, the parasite cells are here, these spheres and clam tissue. This is a section of clam tissue. The animal can mount an immune response. We see it here as encapsulation of the parasite. And that immune response can be effective, leading to healing. The clam can heal, but if the immune response fails in limiting the infection can really spread throughout the body of the clam and kill the clam. So we can have really two outcomes. And nowadays, I think we have a good sense what can lead to one or the other of those outcomes. Looking at the geographic distribution, the disease really, it's limited to the Northeast indeed. The furthest south location was in Virginia, and that's been epizootic, really not many disease events in Virginia. It's been several years now without any disease problem down, and never further south, while the clam can go down to Florida. So the clam can go down, the disease limited down to Virginia. That can indicate already that temperature may be a limiting factor. In New York, so really the disease started here in New York. The first discovery of the disease was in 2002. And that led to a monitoring program surveying the area where the disease was initially discovered here off Staten Island, the area we call Raritan Bay. So the bay was immediately kind of divided to different areas to facilitate monitoring and fisheries management as a result of monitoring investigation. So monitoring of disease in this area in Raritan Bay, different sites. You will see some of these numbers later on the next few slides. And this is one of the slides. So it shows those areas, each area here, a number on the x-axis. What I want to highlight here is that there is a gradient. This is worth of 10 years of monitoring. So it's really, each bar represents several hundred clams, really, if not thousands for some. But the idea here is that the disease can be extremely low in this enclosed embayment here, area 20, where clam density is high, is extremely high. So it's not a matter of clam density in this particular case. Disease is also low here in area number 1. So these are really the two lowest spots for disease in the whole bay. I should say that here we have the Raritan River goes here on the left-hand side here to the west of area 1. Okay? Raritan Bay, which is an important river, so freshwater discharge. This is area 20 is enclosed again. It's a harbor actually, so enclosed, relatively shallow. We'll get back to that later. So that's already indicate possible environmental condition as possibility salinity for area 1 and possibly as temperature for area 20. And this is what we see indeed, at least for temperature. This is area 20. The yellow here is area 20. We have almost 2 degrees difference on average with the remaining areas of the bay where disease is heavy. So the lowest disease we see is in this embayment that have the highest temperature of all. And this is something I have studied also Soren Dahl, who did a wonderful work analyzing this large set of data over 10 years of worth of survey, and clearly demonstrated, I think, that a negative relationship between summer temperature and disease prevalence, and positive relationship between salinity and disease prevalence. I should say that, again, each point shown on this graph represents several hundred clams, so it's not one clam, or so it's really robust statistically talking. So we can look at the data in different ways. Here he used summer temperature and weighted histological prevalence. Here salinity and weighted histological prevalence. But also he looked at peak high temperature each year, also showed a strong negative correlation with disease, and looked also at precipitation and snow. So overall, freshwater inputs to the embayment, to the bay there, Reddington Bay, and have also a negative correlation with precipitation. So high precipitation means low salinity, obviously. And looking at both salinity and temperature, he was able to really find up to 81% of the variance of the data was explained by salinity and temperature, and clearly high disease levels, with high salinity and low temperature. Again, temperature the lowest is here. So basically, in this particular bay, Reddington Bay, disease is heavy right in the center, while refuge area, somewhat refuge, not completely refuge, somewhat refuge area in 20 is a good refuge area, I think, and area 1 here at near the Reddington River, while the disease is heavier in the center. And these findings, actually, temperature and salinity, are in perfect match with the trends we see in lab experiments, when we show high disease development in laboratory studies with high salinity and low temperature. This is for salinity here. This is for temperature. So finding in agreement with lab studies. So both temporal and spatial analysis for this particular case suggest that warmest seasonal temperature restrict infection and areas subject to low salinity may serve as a disease refuge. And I should remind you that the trends for temperature in our region, this is out of south shore of Long Island, Noah Buoy. The trend is obviously on the rise, like throughout the Northeast. There is no exception here. So the idea is that if the trends continue, at least for Quebec's disease, the situation will improve for that particular specific infection. So I will end here. Really, I already used my 50 minutes, I think. So conclusions are really short outlook. For lobster, southern New England statute is very alarming. Management action may help in the south, but in particular in northern locations. I'm thinking here Maine, obviously. For the oyster, there is big concern for wild stocks, but the good thing is there are a lot of effort. We can produce oyster in hatchery setting, and we can really contribute to improving resistance of oyster to infectious disease, but also to environmental conditions through development of resilient aquaculture strains for food production and for restoration as well. For the surf clam, there is still lots of unknown about the different genetic structure of so the genetic structure of the population, whether we have subpopulation that are more or less susceptible to temperature or not. But in any case, there is a good effort. At least there have been a paper published 2 weeks ago about the usefulness and ways to improve management for the species. So the bright spot I mentioned voluntarily should not really hide the big concern we have for shellfish resources in the Northeast, as you saw throughout this talk and throughout other talks you have seen on this series and elsewhere. We have several concerns with the climate, and obviously the ocean acidification is a big ticket item there. There is lots of unknown, and again, we may have good knowledge of the effect of acidification on a species, but not necessarily on that species in relation to other species that interact with it, whether host pathogen interaction, predator-prey interaction, and so on. So it's really complex, and there is really a lot to do and a lot to learn out of that. With that, I would finish really with the acknowledgement. So for what I presented today, the most important sources of funding really were the New York State DEC and New York Sea grant. Some of the results were covered partially by some NSF grants we had, as well as USDA and the Northeastern Regional Aquaculture Center. The lobster work was funded by NOAA and MFS and DEC, New York State DEC. We work heavily with the industry, with the agencies, and we are very thankful to their assistance with the biological samples. Several colleagues contribute to this work, and lots of students and staff here at the lab who really help with processing thousands and thousands of specimens. With that, I end. Thank you.
Excellent. Thank you so much. We have time for one or two questions, and we have one that has come in so far from Joseph Kunkel. What type of management actions are feasible to help the lobster status?
The lobster, excellent question. I should say that to my defense, I'm not a fisheries biologist, right? I'm more a health and physiology person. Nevertheless, I think understanding the distribution and the population density hotspots, possibly areas more protected than others from environmental stressors, I'm thinking. And Maine, again, my thinking is for Maine. Fisheries pressure, this is an area where we can work on it, I think. If some places we know the environment is a little better than others, we can have better protection for those places. I'm not thinking spawner sanctuary for lobster because we know how much they move, but managing fisheries and understanding already the distribution and the environmental quality of the various places where lobsters are present. That's a good start, I think.
Thank you. We also have a comment from Esperanza Stanchoff. She says, nice presentation. And I have a question. So if I understood this correctly, when you were talking about the CO2 exposure to both oyster and hard clam larvae, it seemed that the oyster larvae were more susceptible to bacterial infections after CO2 exposure than the hard clam larvae were. And I wondered if I interpreted that correctly, do you have any hypotheses about why that may be?
In reality, Meredith, oysters in general are more susceptible to infection than clam, and whether adults or larvae, frankly. So this may be part of that. Nevertheless, I need to go back really, we need to be careful if the age was similar, because even a few days, you know, if we are talking 10 days old versus 14 days old, that's already 40% difference, yeah, in age.
Yes.
So we need to be careful. Frankly, I forgot what was the exact age. I can go back to the slide, but we need to be careful about the exact age of the larvae. And again, two species, two spawning events, there is some variable here. So, but overall, I go back to my initial comment. Oysters in general are more susceptible to infection than hard clams.
Okay, thank you. And a question from Jason Masters. Have you looked at the Atlantic multi-decadal oscillation as a temperature factor?
Yeah, I didn't do it myself, although I have some colleagues who did some of that. And we just received funding from NOAA. Actually, I should not say NOAA. It's New York State funding that was funded through New York Sea grant to look at some of that as well. So we will be considering an environmental factor in a broader sense.
Okay.
If that answers the question.
Yes, I think it does. So we are at 3 o'clock and we don't have any more questions right now. So Bassem, I thank you so much for this really interesting presentation. The recording of this presentation will go on the NECAN website. So if anyone has colleagues who were not able to tune in for the live webinar, they will be able to see it once it is uploaded. So thank you so much to all of the attendees and to Bassem today. Thanks.
Thank you all.
Bye.